Okay, so this is the first of a series of detailed videos about each system that you need to know for this for chapter 6. So I'm going to go into just a little bit of detail, just kind of going over some of the content again. And so we might as well get started. So the first system is digestion. Digestion. This is 6.1. And the first thing that you need to be able to do is explain why digestion of large molecules is essential. So in other words, um, we're asking the question of, so why is digestion required? Essentially, that's what we're asking: is what's so important about digestion? And really, I guess what we need to know is we need to be able to define the word digestion, and this just simply means to break down large molecules. We got to be able to break down large molecules, and the main question here is, why do we have to do that? And there are two key reasons. The first is that so some large molecules that we eat are not usable to us. So what we have to do is break them down and then rebuild the way our body needs them. So that sounds very vague, and sure, you can know that, but do you really understand that? That's a different question. So what I'll do is actually give you a more concrete example. Now, this requires a little bit of knowledge about ecology, but if you think about food chains, which is a simple model. If you think about, and I won't use technical words here because we're just using this for conceptual understanding. Producers, think of plants, analogy that do photosynthesis, they're called producers. And then they get eaten by consumers. Really, we'll call them primary consumers, secondary consumers, and on, right? And then maybe it goes to decomposers. Some kind of bacteria or something, or fungus that decomposes. In actuality, this will go here too, but now I'm starting to draw a food web. So if we think about it in this way, producers make food, make certain molecules that the primary consumers ingest and then digest. So they break down into small molecules, they rebuild it the way they need it, and when the secondary consumer eats that primary consumer, it breaks down those molecules into the small pieces and rebuilds it the way it needs to. So a good example of this that I often talk about is protein. So these producers have protein. Plants have proteins. When the primary consumer eats those proteins get digested down into amino acids, and then within that primary consumer, the amino acids get put back together by ribosomes in the way that the organism needs it to make proteins. And then when the secondary consumer eats those proteins, the secondary consumer digests that back to amino acids, and the secondary consumer builds those amino acids up into proteins. So this cycle goes over and over and over again, and I kind of think of it as a set of Legos. A kid might have a set of Legos, build something. When playtime is over, takes those Legos pieces apart back into the individual pieces, puts them away, and then the next time the kid comes back, takes those same pieces and builds something new out of them. And that's all that we're talking about here. So some large molecules we can't use in our bodies, so we have to break them into the small pieces and put them back in a new way that our body does need. So this is the first reason that we need to break things down. We have to digest things. The other reason is that these molecules, in order to pass cross the membrane, cell membrane, of the villus of the small intestines, molecules must be small. They have to be small enough to go through the membrane. So that's quite important for and a good explanation for digestion. But while we're on it, let's talk about a couple other words. Digestion, we said, means to break. So I can spell and talk at the same time. Digestion means to break things down. Ingest means to put something in your body, so in your mouth. We've got absorption as another term, and assimilation. And there's so ingestion happens first, then digestion, then absorption across the villus, and then once those small pieces of or small molecules make it into your bloodstream and around your body, into the cells put them together in the way they need to. That's assimilation. So when those amino acids get put into proteins, that's our example of assimilation in this case for proteins. And so the other thing you should also focus on is what's the relationship between monomers and polymers. Proteins, another word for proteins is polypeptides. These proteins are a polymer. They're made out of these building blocks or monomers called amino acids. So you can tear them apart into the building blocks or put those building blocks together to make the polymers. Okay? Okay, so let's move on. So we're talking about digestion. And let's talk a little bit about enzymes. So enzymes, just so you know, are a type of protein. Not all proteins are enzymes, but it's a type of protein. So just so you know, we already know this because I just talked about it, made of amino acids. And what enzymes do is lower the activation energy or the energy required to drive a reaction forward. And by doing that, they speed up reaction rates. Or in other words, they are catalysts. They speed these reactions up, and they are reusable unless they are denatured, which means they change, that's delta, change shape. And the way that they become denatured is when the pH changes drastically or temperature changes drastically. So those are the two key ways. There's some other ways that these proteins can be denatured, but those are the two that we're going to focus in on. What we should know is that these enzymes break down large molecules into smaller ones. That's the reaction we're talking about. So they speed up the reaction, and they work to help break down or digest large molecules into small molecules. And what we should know, we'll talk about pH in just a minute, but the optimal temperature for all enzymes in the human body is about 98.7 degrees 98.6 degrees Fahrenheit. But because this is science and we use metrics and there's IB and all that good stuff, you should also be familiar that your body temperature is about 30 degrees Celsius. And that's the optimal pH of these enzymes and where they function best. But pH is a little different. So let's go and talk about pH, which is the next thing. You need to be able to say the source, substrate, products, and optimal pH for one amylase, one protease, and one lipase. Okay, so make a little chart here to make sure we have everything that we need. So our example of an amylase that we talked about in class is salivary amylase. The example of the protease that we talked about is pepsin, and for lipase, we can know it as the pancreatic lipase. And now I know this is a point of contention on a quiz, so I went back and readjusted grades, so don't worry about this. This is what you need to know. I double checked. IB wants you to know the pancreatic lipase is our example here. All right, so the source for the salivary amylase is the salivary glands. The source for protease is the chief cells. That's what they're called. Chief cells in the lining of the stomach. And lipase, pancreatic lipase, the source is the pancreas. Okay, so the substrate is the molecule that the enzyme bonds to, that it helps break down. So remember we talked about these enzymes in particular help break down or digest bigger molecules. So we're talking about what big molecule do these things attach to. And the substrate for amylase is starch, also known as amylose. For the pepsin, all you need to know is protein. It's a generic term, but you might have heard of this word, so you could use this one as well, polypeptide. And for lipase, these are lipids, but specifically triglycerides, like oils and fats. Okay, so you need to know the substrate and then the product. So the product of the reaction, remember enzymes help drive reactions forward. So what enzymes do is takes the substrate and breaks up into something smaller. And so what salivary amylase breaks the starch down into is maltose. And we're going to talk about this in the next unit, but starch is a polysaccharide, maltose is a disaccharide. The protease breaks the proteins into smaller polypeptides, and sometimes even individual amino acids. And the lipase breaks these fats down into fatty acids and glycerol. Now the optimal pH, remember we were talking about denaturation earlier, the optimal pH is the pH at which this reaction works best, and the optimal pH in the mouth, or where the salivary glands are, is 7. The optimal pH for the stomach, where pepsin is 1.5 to 2, and the optimal pH for the pancreas is 7. So notice that as food travels through the digestive system, it's neutral, highly acidic, and back to neutral. And these are the optimal pHs for these enzymes in these places. A good way to remember the stomach is acidic is, I think you know that there's hydrochloric acid in the stomach. This is a strong acid. That's what these numbers indicate. This is a strong acid. And so this hydrochloric acid serves a couple functions. One, to keep the pH low or highly acidic so that the pepsin works efficiently. It also kills bacteria that might enter our system through the digestive system. All right, so the next thing that you need to be able to do is draw the digestive system, which I'm not going to do because there's another podcast on drawings. So let's talk about, you have to know the key functions of some organs in the digestive system. So let's talk about, so you got to know functions of the stomach, the small intestine, and the large intestine. Now, of course, you need to know the functions of all the parts, but these in particular. I just mentioned this. So the hydrochloric acid in the stomach kills bacteria most of the time, unless there's some bacteria that are resistant. The hydrochloric acid provides optimal pH for enzymatic digestion. And number 3, it secretes pepsin, which is the enzyme. Okay, so those are the functions of the stomach. The small intestine. So the wall actually, interestingly, secretes enzymes. It receives additional enzymes from the pancreas. And lastly, has villi to aid in the absorption of nutrients. The large intestine moves undigested food and unabsorbed food out of the system. It also, not remove, but absorbs water. And third, produces feces. So you need to know this chart. Really easy to memorize and reproduce. Okay. So almost done here. So you're going to talk about absorption versus assimilation. We already mentioned this. Absorption is moving small molecules across the membrane. And assimilation is when cells use the monomers that were absorbed into the bloodstream to make new polymers to be used by the body. Okay? Alright, so the last thing here, the last bit for digestion that you need to know for now anyways, let's see if I've got any room. Yeah, okay. Is the structure of the villus and its role in absorption and transport of products through the from the digestive system. So I'm going to draw a villus. That's not a very good drawing, but a villus here. And that second line should be as pretty close to the first one. And I'm going to draw cell membranes across here, so we see that the thickness of this membrane is only one cell. I'm going to label this as the space within the tube called the small intestine. This is the lining of the small intestine. This is a villus. This whole thing is right here. And the villus has a capillary bed that runs right along the inside here. So that as food passes through the small intestine, monomers that have been digested can diffuse across the single cell membrane into the bloodstream. But there's one exception, and that's the lacteal, which is one tube that is bigger and sits all the way in the middle of the villus. And what it does is transports lipids, because lipids are often bigger than carbohydrates and proteins. So here's the blue thing is the lacteal, the red is the capillary bed. And so we have to talk a little bit about how this is modified to increase absorption. And so here's a list of things that you should know. So there are many villi which increase the surface area to volume ratio, which increases the ability to absorb things quickly. The membrane is only one cell thick. Third, the villi on the ends here have little hairs, and these are microvilli. So microvilli increase the surface area even more. Fourth, I said things diffuse across the membrane, but in that membrane there are also protein pumps and channels that speed up absorption. Fifth, there are mitochondria that sit inside these epithelial cells or the membrane cells that produce ATP or energy molecules to fuel the protein pumps. And six, that the blood capillaries are very close. So let's say and then seven, the lacteal takes away fat. Okay? So that should do it for digestion.